So now we're going to talk about regulation of our TCA cycle. So there are some pretty general regulation mechanisms. So some of these we've already discussed. You can have isozymes of enzymes, and so they can have tissue-specific differences where you'll have an enzyme in one tissue that has a high affinity for a substrate, and the enzyme in another tissue will have a low affinity for the same substrate. So that's one way to regulate enzymes. And then you can also regulate enzymes by having a slow step or a rate limiting step. Oftentimes these steps are also called commitment steps, and they can occur at a branching point, which means if you want to go in this direction, you have to make sure you want to be in that direction because I can go a different way as well. So it's sort of like there's multiple options at that branching point, and when you go in one direction, that becomes a commitment to whatever process that is. Oftentimes you also have feedback regulation by some sort of end product. The end product can be at a different branching point, or it can be the actual end product. Another way to regulate genes is through compartmentalization. This is, for example, keeping our TCA cycle enzymes in the mitochondria, keeping lysosomal enzymes inside of a lysosome. So you're able to control the function of those enzymes by controlling their location. Also, you can have integration of messages from the outside controlling enzymes. So hormonal regulation through phosphorylation, which is a very easy way and quick way to turn on and off enzymes. The amount of enzymes, so some of these hormonal regulations will cause an increase in enzymes being transcribed and translated. And you can have concentrations of activators or inhibitors in order to control an enzyme. Now, as far as the TCA cycle, we have to start first with our regulation of your pyruvate dehydrogenase complex. And so our pyruvate dehydrogenase complex has two forms. We have the inactive form and we have the active form. Now, of course, in the active form, you're going to go from pyruvate to acetyl-CoA. And in the inactive form, you're not going to be able to do that. So what is driving the pyruvate dehydrogenase complex? Well, there are certain things that are driving it. A need for energy, calcium is a positive regulator of your pyruvate dehydrogenase complex. Whereas you have acetyl-CoA and NADH is going to be a positive regulator for a kinase that is going to add a phosphate and inactivate your pyruvate dehydrogenase complex. So in the case of pyruvate dehydrogenase complex, phosphorylation of the complex leads to inactivation. Dephosphorylation by a phosphatase leads to activation. And so it all is going to be about need. Do you need to make energy or do you have too much energy? So again, if you have acetyl-CoA, which is going to feed into the TCA cycle, that's a negative regulator for the PDH, and that is going to activate the kinase to phosphorylate. If you have ADP and you need energy, that's going to turn off the kinase and is going to push it towards an activated state. Okay, so there's many points where we can actually regulate our TCA cycle. So at the level of citrate, which is the first commitment state, it is going to be regulated negatively by citrate. For our isocitrate dehydrogenase, we're going to have negative regulation by NADH. For our alpha-ketoglutarate dehydrogenase, NADH is negatively regulating. For our malate dehydrogenase complex, again, a negative regulation by NADH. So you can't make more NADH if you run out of NAD. And so in order to recycle NAD, you have to donate those hydrogens to the electron transport chain. Now positively, you're going to have regulation of isocitrate by need, a need for ATP by having higher levels of ADP, or driving it by calcium, such as in muscles where they release calcium through the sarcoplasmic reticulum. Calcium is also going to push through from alpha-ketoglutarate to succinyl-CoA. So citrate synthase is pretty simple. It's going to be a feedback regulation. So again, citrate synthase is going to negatively regulate, is going to be negatively regulated by citrate. And another side over here is malate. So we have malate and oxaloacetate. Malate is going to favor, the formation of malate is going to be favored over oxaloacetate. Remember, malate can leave the mitochondria. Your oxaloacetate cannot. Citrate can leave the mitochondria. And remember, citrate is a negative regulator of glycolysis. And that's through PFK1. Oops. Phosphofructokinase K1. So citrate, if it builds up, you're going to have to get these carbons out of the mitochondria. And so citrate will actually leave and will start to block glycolysis because you don't want to keep pushing pyruvate through to make acetyl-CoA if you have a backlog of citrate. Now, NADH, NAD ratios are very important. Again, in order to make NADH, you need NAD. If you have too much NADH, that means your NAD is favoring NADH, so you have a high ratio. So if the ratio decreases, you're going to push towards oxaloacetate. If the ratio increases, you're going to push toward malate. So we also have alpha-ketoglutarate and isocitrate dehydrogenase that we just mentioned. And again, they're going to need NAD plus in order to push these forward. So if they have a lot of NADH, they're going to negatively regulate these enzymes. Calcium, again, is an obligate activator of these enzymes because it's telling the cells that you are doing some sort of activity and you're going to run out of energy pretty quickly, so you need to start making it. ADP also, levels of ADP increasing will push isocitrate to alpha-ketoglutarate. Now, just as a side note about regulation, we also have our nuclear proteins. So the mitochondria does have their own DNA, but the mitochondria relies a lot on cellular DNA. So in the cytosol, you're going to have proteins encoded from the DNA that are going to be translated on free ribosomes. So you have our mitoproteins, and these are made on free ribosomes. So these do not go into the ER. They're not associated with ER. Now they are going to be associated with heat shock protein 70. So that's shown here. We have our heat shock protein 70 that will pick up these proteins that are necessary to enter the mitochondria. Now all of these are going to have complexes that allow it to cross your proteins. So you have a outer membrane complex. It's a transfer protein outer membrane. And you have an inner membrane, TIM, a transfer protein inner membrane. And TOM is going to be made up of proteins that are going to be able to bind to a pre-sequence. So these proteins here will have an N-terminal pre-sequence. And this pre-sequence is going to be able to bind to TOM. And this will facilitate its movement into the inner membrane space through the outer membrane. Now the TIM complex is going to allow these proteins to get access to the matrix of the mitochondria. And so here's the matrix, here's the inner membrane space. You're going to have insertion of this pre-protein into TIM, and this is driven by the potential difference across the membrane. Now, heat shock protein 70 binding to tin complex is important in this process. So heat shock protein 70 is going to facilitate the transfer of these proteins into the matrix of the mitochondria. Now, once they're in the matrix of the mitochondria, these proteins are often going to need to be folded, and heat shock protein 60 is going to help fold the proteins, or the enzymes. And so the final step is going to be cleavage of the signal sequence by the matrix processing protease. So they're going to lose that N-terminal signal sequence in the mitochondria matrix.